In this series, I'm going to talk about FTIR as a tool for the characterization of sensing elements of chemo or biosensors. So let me tell you that the learning objectives of this talk is going to be basic principles of FTIR. So you learn what are the basic information, what is the basic principle of FTIR is. Then you will know what are the information which can be obtained from FTIR. So once you learn the principle, you will know that where you can apply it. Then you will also learn that out of the several techniques you have learned, what are the advantages of FTIR? What is the basic instrument? So when you look at when you use this FTIR, you should also know what is inside the FTIR instrument. So we will get some information about that. Then you will also learn about the characteristics of IR spectrum. So you will get to know how to analyze your spectrum, what changes this IR spectrum. And then we are also going to talk about factors which are affecting IR band. Next thing which we are going to talk about is FTIR as a tool for characterization of nanomaterials. This is one thing which is very important. That is why you are attending this, seminar, where we are talking about how you can characterize the various nanomaterials as well as various sensing elements. So suppose you have deposited a thin film and you want to understand what is the material which is deposited over here. This is the tool which can give you information about that. Uh, okay, so now let's start with FTIR spectroscopy. FTIR as such stands for Fourier transform infrared. The preferred method of infrared spectroscopy is FTIR. That means, do we have another way of looking at infrared spectroscopy? Answer is yes, you can do the measurement, but I will tell you what are the advantages and what are the disadvantages of that. In infrared spectroscopy, IR radiation is passed through a sample. In both the cases, you will find that IR radiation is passed through the sample. And when some of the infrared radiation is absorbed by the sample and some of it passes through it. So that means it is being transmitted. The resulting spectrum represents the molecular absorption and transmission, creating a molecular fingerprint of the sample. So for each molecule, you will have very specific fingerprint. So like a fingerprint, no two unique molecular structure, they will produce the same infrared spectrum. This is why you will find that each molecule will have a very different spectrum, and that's why you can analyze them. So look at here, it is a source, then you have infrared light, you pass through the sample, and the energy which is shown over here, you will see that some of them, they are being absorbed and rest is being transmitted. So you are collecting the data based on the transmission, and then it goes to the detector where you analyze the data. Now here, FTIR spectrometer is slightly different than the other spectrometer, and that is what we are going to discuss, the physics behind it. So in a typical frequency domain, in general, the optical spectrometer, the radiation from a source that enters through the monochromator. So in most of them, you will find that monochromator is a basic ingredient or basic component of your spectrometer, where what you will find is that particular wavelength or frequency is selected and then allows to pass through a sample of analyte. So you have an analyte sample in a cuvette, and then it is being allowed to pass through after it is coming from the monochromator. So it is selecting the specific wavelength, and that is what is being passed through. But there is another. So other than your frequency domain, you also have a time domain. In time domain, what you have is that the pulses the sample with a signal containing the entire range of frequency. So instead of selecting the frequency, entire range of frequency at once is being passed through the sample. So information contained is same. So in case of whether it is a frequency domain or it is a time domain, you get the same information, but there is a difference. So what is the difference? See, all the data is collected in a single observation, and it is a very fast than collecting one frequency point at a time. So whenever you are collecting the data in time domain, you have advantage, and this advantage is that you are collecting all the data in a range, and it's a very fast, and that is why you have resolving power between peak is often much better than you collect in normal spectrometer. Now what happens in FTIR? You are collecting the info the in the time domain. But after collecting the data from time domain, what is done in FTIR instrument? It records the signal in time domain, but what it does is that it turns the signal recorded in time domain into frequency domain. So basically, the role of Fourier transformation is to convert it from time domain to frequency domain. Now, looking at in which range we have infrared radiation, this is a very common thing which you all might be knowing, that infrared radiation, it lies between visible and microwave portion of the electromagnetic spectrum. So if you look at the electromagnetic spectrum, it is a microwave, and this is the the visible range. So between visible range and microwave, you have infrared range. Here you have a near infrared, mid infrared, and far infrared. So all this range is between the microwave and visible light. Based on IR absorption by molecule as it goes through vibrational and rotational transition, uh, we need to understand what is happening when you pass or flash infrared. What is going to happen here? So basically what is happening is that when IR is absorbed by the molecule, it undergoes vibrational and rotational transition. So these are two transitions which has been shown in terms of their energy. So either you can have rotational type transition or vibrational transition. Absorption of radiation in this region by a typical organic molecule results in, you know, these two kind of, excitation where the rotational transition or vibrational transition occurs. And one more type of transition which occurs is a bending. That is what happens in this process. So rotational transition have a smaller energy difference compared to vibrational transition, which occurs at higher energy. Now, rotational and vibrational transition often occurs together. So it's the combination effect which you see at the end, because of which your spectrum changes. So why we should go for infrared spectroscopy? It is because infrared spectrum, it is representing a fingerprint of the sample. So you have seen that each molecule will have different. So I'll show you that even if you have the same number of a carbon, same number of a hydrogen, and you have the molecule that differs in terms of their double bond, or differs in terms of their triple bond, or a single bond. So you will find the difference in their fingerprint. So because each different material has a different combination, even the combination of the atoms, that can give you a difference. The presence of double bond can give you a difference. So all these things are counted when you are looking at the spectrum. Now, this is an example which shows that how the molecular vibration or bending is occurring. So here you have mainly two phenomenon which is occurring. One is stretching, which could be symmetric or asymmetric stretching, or it could be bending. Here you can have different ways in which the kind of molecular vibrations are happening. Why it happens? Because the position of atom in the molecules are not fixed, and that's why they keep on showing this phenomenon. It corresponds to absorption energy of a molecule, and the one you have seen is basically stretching mode and bending mode, which has been shown over here. Now, this is a typical spectra of IR. So those who have taken the IR spectrum, they know about it. Otherwise, this is the way you get your data. So what is this data reflects? For example, the top one, this is one showing the transmission pattern of the hexane. So what you find here is that there are specific transmission transmittance spectra where, for example, this is the peak, these two are peak, and then this is the peak. So there is a characteristic of this molecular organization of hexane. So it is having 6 carbon, and that's why it is a hexane. And what you find is that as you can keep on having, same number of a carbon atom, but what you have is that you have double bonds. So because of the double bond, what you find is that there is a pattern which has changed. You will get additional, transmission peak and transmittance peak, which is very important for your, spectrum. So this transmittance data, you can have either in the transmittance form or you can convert them into absorbance form. That doesn't matter. The point is that even a molecule differing in presence of the bonds, that can be characterized. So here, this is a typical example. It is a 6 carbon compound having no double bond, having a single double bond, or having a triple bond. So because of the presence of the bond, you have seen that how the spectrum is changing. Now, how we calculate the vibrational mode of these, molecules? So, for a non-linear molecule, the number of types of vibration which can occur is 3n minus 6, whereas for a linear molecule, number of types of vibration could be 3n minus 5. So that means linear and non-linear molecule, they will show a different types of vibration or different number of vibration which you can get. So observed vibration can be less than the predicted one. So this, the one number which I'm giving, this is the predicted one. But of course, when you go for taking a reading, there may be number could be lesser. The symmetry, when there is a no change in dipole, energies of vibrations are identical. So these are some important facts which you need to know. And absorption intensity is too low, and frequency beyond a range of instruments. So these are the things which you can monitor when you are taking the actual reading. And because of that, the observed vibration can be less than the predicted vibration. So what could be the predicted vibration? Predicted vibration could be 3n minus 6 if it is a non-linear molecule, or it could be 3n minus 5. Now the example of calculation is, for example, you have HCl, which is having, a linear, which is a non-linear, Cl is a linear molecule. Here, what you get is that you apply the formula of 3n minus 5. So you can see the number of mode could be 6 minus 5. So you will get only one mode of vibration. But if you take CO2, in CO2, you will have, 3n, n is 3 because you have COO, and that is why you have 9 minus 5, that is the 4 modes you can observe in case of CO2. So this gives you a predicted way of calculating the number of types of vibration. Now I will give you some very interesting fact that you can have IR spectrophotometer, which could be dispersion spectrometer, or it could be FTIR, which is a Fourier transform spectrometer. So what is the difference between these two? When you go for a dispersion spectrometer, you have light source, same thing is in FTIR, but the difference occurs here is that here you have a grating, and because of the grating it will separate IR light, and then it passes through a slit, and then the light passes through the sample and then finally reaches to the detector. So what you are doing is that here the process is involved where you are selecting a specified IR light of a particular wavelength range. So in this process, what happens that it takes several minutes, number one. Number two is that the energy which reaches to the, detector, starting from the light source to the detector, is very few percentage which is going to reach over there, because what you are doing is that you are passing it through the slit, and you are also selecting a particular wavelength range, or a particular wavelength, and that is what is passing through the sample. On the other hand, when you take Fourier transform infrared spectroscopy, what you are using is that you are using a different method, where I will describe it a little later. It is basically an interferogram, and it is it is based on interferometer, which is using IR light, and then it is passing through the sample. So what you are looking at is that lot of energy which is coming from the light source is passing through the sample, and that is why here it measures, it takes very very few seconds for monitoring the spectra. And second thing is that detector receives up to 50% of the energy of the original. So a lot of energy is being received, and that is why you find that it is a very very sensitive method. So what information one can obtain from FTIR? First thing is that you can identify unknown material. It can also determine the quality or consistency of a sample. Other than that, it can determine the amount of a components in a mixture. So these are the information which you can obtain from FTIR. Now let us look at the advantages of FTIR. So Fourier transform infrared spectroscopy, or FTIR, is a preferred over dispersive or filter methods of infrared spectral analysis because of several reasons. Number one, it is a non-destructive technique. It provides a precise measurement method which requires no external calibration. So it's very important. You are not calibrating it. It can increase the speed, collecting a scan every second. And so that means whenever you get one spectra, it takes very, very less time. As I told you in the previous slide itself, that it is a very fast technique, and it can increase the sensitivity. 1 second scan can be co-added together to ratio out random noise. So what you are doing is that you are reducing the random noise. Uh, so what it means, for example, you are taking the spectra of IR spectra using FTIR. So whenever I put it, I will put it for 3 minutes. Now in 3 minutes, you will find that the spectra is recorded by the system, say something like 256 scan. So normally when you use this FTIR, you fix the number of scan you want. So that means that many times your spectra has been collected, and then it is co-added, and this is where what you can discriminate is between the real spectra than noise. So that is what is the greatest advantage of using FTIR. It is mechanically simple with only one moving part. So that's another important thing which you find in FTIR. This is a typical instrument. If you come to the lab sometime, you will see that this is the spectrometer. It has different parts. It is not shown, but the internal part, which is available in this inside the instrument, this is given over here. So typically, what you have is that you have IR source. There are various mirror, beam splitter. You have interferometer. You have again several types of mirror, then it goes through the sample. And this is a sample compartment. We will talk about that the sample compartment can be changed. You can have different types of samples. And so this gives you a basic design of the FTIR, where you have a source, you have a laser, you have interferometer, and finally you have a detector. There's another, instrumentation which is shown the internal line diagram, which shows that how the organization of different parts of this is possible. And in a typical instrument, not actually FTIR, it could be, the way the overall, process can be collected. So now coming to the source, there are different types of source of light which is used in case of IR spectrum. It could be NOS Glover, or it could be laser source. Now what happens? Now this is a very interesting thing which you find over on the right hand side. So this is the one which is showing you how interferometer works. It is a typical interferometer, which is a Michelson interferometer. So here what is happening is that it is a device. So interferometer is a device in which you are having two or more radiation beams that interfere. So that's why you have this kind of part in the system. And what is happening here is, so suppose you have coherent light source goes, and then you have a mirror, it is going to, reflected, then another mirror, this coming back here, then you have a third mirror, and finally from here it goes to detector. So there is a set of mirror which is involved in the interferometer, and that is why what you see is that you find that your beam is being split. For example, here is split on this direction and this direction, but ultimately all the beams are coming back, and they are they're different paths, and then finally it combines and goes to the detector. So then this Michelson interferometer, it recombines the two beams as you have seen here, and then where the difference of intensity of these two beams are measured as a function of difference in the path. So you have seen there are different paths, and that is what is going to be measured. It produces unique type of signal which has all of the infrared frequency encoded. So instead of having selecting a particular frequency, what you are doing is that you are sending all the frequency, and that is what is going to be recorded. Now, it modulates the IR signal at lower frequencies so that it can be observed by the detectors. The major job of this interferometer is that it is going to modulate your IR signal. And then finally, the two domains, they are going to be interconvertible by a mathematical method of Fourier transformation. Now, coming to the detector. So I'm talking about the instrument where you have learned about what could be the light source, what could be the, now we are going to talk about the detector. Detector could be thermocouple, bolometer. So there are different types of detector. I will just focus on those detectors which are commonly used in the current day, uh, IR spectrometer. So for example, photoconducting detector, pyroelectric detector. So there are 3-4 types of detector which is known for FTIR, but most commonly used detectors are more sensitive detectors are DTGS detector and MCT detector. Now, the question comes, which detector one should use? Which detector you are going to employ in your instrument? Or suppose you are purchasing an instrument, which detector you should select? So here, DTGS is the one which is a very sensitive at room temperature detector. And it is for mid-infrared range measurement, and employs temperature sensitive. And so the basic thing is that it is going to employ temperature sensitive ferroelectric crystal of deteriorated triglycine sulfate. So this is the one which is making your detector. And as the temperature and hence polarizability of the crystal changes, why should it change? When infrared excites this, and when infrared is absorbed, you will find this change. And it is generated which can be detected by two parallel electrodes. So this is the way your DTGS detector works. Another very important detector is a MCT detector, or mercury cadmium telluride detector. It is a sensitive for very sensitive work. For example, if you're working in the protein spectra or biological samples, and you want to further detect the signal you have obtained, and you are going to further analyze those spectrum, you need to have very sensitive detector. But the like DTGS detector, which can be used in room temperature, this MCT detector requires cooling, and that's very important component. So if you need high sensitivity, your detector requires cooling, and that is where the detector has to be coated to liquid nitrogen. So what you do is that during the time of experiment, you fill them with liquid nitrogen, and that's why the for example, couple of hours it will remain cooled, and that is where you can take your spectrum. The output from detector goes to preamplifier, where it is converted into voltage signal with varying with the time. So that is where you see. So these are the two different modes in which sample preparation is done. Now, this is the one where you use infrared transmission spectroscopy. Transmission means that it is passing through the sample, and after passing through the sample, certain light is being absorbed and rest is transmitted. So that is what is the infrared transmission spectroscopy. What you do here is you take the transmission of the solid, some of the solid which can transmit them, transmission of liquid, it's a very common, and transmission of a gas. On the other hand, suppose you have a very thick material. What you can do is that you can use attenuated total reflectance, and that is why I'm talking about this. For suppose you have deposited a film, and which is thick film. So under those conditions, you have to use ATR mode or attenuated total reflectance mode. Suppose you have a crystal and light cannot pass through that, you can go for ATR mode. So there are different infrared reflectance spectroscopy which can be employed here. So you have attenuated total reflectance, you have diffuse reflectance, and specular reflectance. Generally, one can use this ATR mode where you can take the spectra of the solids. Now, looking at the different holders, what kind of sample holders you can use. So, if you have a transmission of solid, you have different types of holders which are present. Here, your solid can be held together and they can be passed through. One of the very interesting thing which is done. Suppose you have a powder and you want to study through the transmission mode. What you can do is that you can make a pellet. So, for making a pellet, you have a press. So, use this press, you can make a pellet, and this pellet can be fixed over here. So you this way you can study the solid material. Other than that, you have a very nice cell where you can put your liquid sample, and this liquid sample you can also adjust the volume by looking at how much space is present in between them, and that gives you an idea that you can load your liquid sample, and this liquid sample can be put in the in the IR spectrometer, and you can analyze. Now for gas, you have a different type of holder which can be used for analyzing. So now coming to the advantages of transmission infrared spectroscopy, you find that it does not require an additional accessory other than the holder. The same instrument, same material can be employed, and it it used to collect used in the collection of many reference library spectra. So let me tell you that for various compounds, we have a library. So it can become easier for you that this is the spectra of, say, hexane, or this is the spectra of, say, this particular organic molecule. And you can compare, you can feed the data, and you can see that whether you are having the same molecule or not. Now, this transmission infrared spectroscopy is ideal for quantitative measurement as well, and does not require extensive training. If you want to use it, it's a very simple process. You can do this. Now, coming to the infrared reflectance spectroscopy, this is another way where instead of the absorbance or transmittance, you are looking at the reflectance. And what happens? The infrared beam goes, an evanescent wave moves through the crystal, and at the end, you will find the infrared beam which is exiting, and that is what you are going to record. On the other hand, you can have also diffuse reflectance or specular reflectance modes where you can use the IR source and can get the data. Advantage of the infrared reflectance spectroscopy, which you have just learned, is that you require a minimum sample, and it is a fast and easy cleanup. So, unlike your cell, you don't have to wash your cell. You just clean the the surface, and it is a very fast. Then coming to the analysis of sample, they can be analyzed in the natural source. That means if you have a solid, you don't have to make them in a powder form. If you have a crystal, you don't have to convert them. So that's where it is a sample preparation is in the natural state. And as I said, it is excellent for thick or strongly absorbing samples. Now, looking at the application in different field and looking at the spectra, as I said, it can be a fingerprint of different molecules, organic molecules, as well as it could be the fingerprint of different types of double bond, or it could be a fingerprint of certain groups. So that is what you can see. You can see the single bond. Here you can see single bond stretching. You can say CH bond stretching. You can see nitrile and carbines are in this range. Double bond is in this range. And then you have certain single bond again over here. So you have a very interesting fingerprint of these spectrum, which is basically your either transmittance, or you can put it in the absorbance mode where you can find out that at a specific wave number you can find this transmittance value. This can also be illustrated in the frequency, depending on the frequency. you can find that OH, NH, then CH, OH. So that way you can find that this is the frequency where this kind of absorbance is occurring, and that is stretching vibration, bending vibration, etc., which is responsible for such kind of a spectrum. Now, as you can see here, it can be applicable in pharmaceutical industry, it can be in forensic investigation, it could be for analyzing polymers, lubricant, food research, quality assurance, environmental and water quality, biochemical and biomedical method, coating and surfactant. Let me tell you, a very interesting thing can be that you can attach this IR spectroscopy with the microscope. So you can also do IR microscopy. And in IR microscopy, what you do is that you focus your beam a particular place, and then you can record the composition. So suppose I have a material which is made up of couple of layers. So each layer can be analyzed. There is no other way you can analyze it other than focusing an individual layer and then finding out their spectrum. So it's a very powerful tool. This is the one which gives you that absorbance frequency or wave number, where this different bands, they are going to have a characteristic absorbance. Higher spectra for biological macromolecules, so it is ranging from chemical molecule to biological molecule. Most common thing which has been studied is about the protein, where you have amide 1, amide 2, and very small signal of amide 3. It is not reflected here, but amide 3 is present over here. Uh, Here, amide 1, amide 2, they are important, but amide 3 is very important because it does not have interference of water, whereas interference of water is great in amide 1 region. Now, looking at the lipids, you can analyze nucleic acid, you can analyze carbohydrates. Each one of them have very specific fingerprint. Now, looking at the IR spectroscopy of proteins, I'm just going to give you some detail because let me tell you that you may have learned about the CD spectrometer, which is a circular dichroism based spectrometer, where you pass the circularly polarized light and then you get the spectra, and that spectra can give you the information about the secondary structure of the protein. Here, you can directly get the information. As I said, you can have amide 3, amide 2, and amide 1 range. And here, you can collect the spectra of a protein. So it's basically what you have is it has absorbance, which is associated with amide bond. So you have different amide bond, and they are showing you different stretching vibration. And then you also have a CO bond of the amide. So you have different kind of a bonds which are present in the protein structure. And why I'm talking about amide? Because all the proteins, they are having polyamide bonds. Normally, in biological sense, it is called as a polypeptide bonds. So these bonds, they are present in numerous number, and that is what can be recorded. So you can see that what are the different information you can get. You can get where you have a beta sheet structure in a protein. You can get the information about the random coil of a protein. You can get which is the one peak coming out of the 3D helix. That means helix pattern or alpha helix pattern. So all the information is, present in the spectra which you have obtained. But one very interesting thing over here is that what you do is that, let me give you some, hands-on information. So when you take the absorbance of the FTIR, you get peaks like this. Now what you do is that you know take this and get the second derivative of the spectra. So when you get the second derivative of spectra, you get the information that which one is corresponding to alpha helix, beta sheet, and random coil. And then you take this information and use this information for taking this peak and deconvoluting these peaks. So basically you use the program where peaks are deconvoluted. And when you deconvolute the peak which you have collected from IR spectroscopy, you get these, dotted lines which are showing that how you can differentiate them as a alpha helix, beta sheet, and patterns, etc. Now, when protein unfolding occurs, you may not get these peaks. So it can be a very interesting tool, especially for pharmaceuticals. For example, any other protein which you are putting on the surface of your, these chips, so or your sensing sensor chip. So that is what can be obtained from the IR spectrum. Now, question comes that what are the factors which are going to affect IR bands? In general, the most important factor that determines the frequency where a chemical bond absorbs is the bond order and the type of atom they are joined together. And the intensity of IR frequency are affected by two primary factors. One is a vibration, stretching and bending mode, and then electronegativity difference. So these two are very important parameters. And in general, greater the change in the dipole moment, a larger the peak intensity is observed. So it is a dipole moment dependent information which you are obtaining. Stretching induces more dipole moment than the bending. So that is why you find that it is the one which is leading to the, the your obtaining the IR spectra. The ability of a molecule to absorb radiation during a particular vibration depends on electrical geometry. So it is the geometry of the molecule. It is the, that is why you have seen that whether it is a linear or, non-linear molecule, that is where the IR spectra is dependent. You have also seen that how these atoms, they are arranged in a particular molecule. You also have seen that it is the dipole moment which is responsible for their, their uh, absorbance or in terms their transmittance. So all these factors, they are affecting IR band. Now I'm switching the gear, and what you are going to learn about is the sensors. Because so far you were learning, and many times you may feel, you may come to your mind that, okay, this is the area where a chemist or a biochemist should be involved. Why should I learn about IR spectroscopy? And here, what you will learn is that FTIR has been used as a very powerful tool in developing chemosensors or biosensors. I'm taking one example. They can be extended to chemosensors as well. So, for example, if you are analyzing your data, when you are using biosensors, where do you use biosensors? Suppose you have to analyze any biological material, you are going to use biosensors. For example, I want to detect urea in the blood sample, I will go for a biosensor. If I have to detect the presence of glucose in the blood, I will go for a biosensor. If I have to detect, for example, the cancer biomarker, then I will go for a biosensor. So biosensors can be applied for detecting any biomolecule for which we need, where we can have a specific signal and which is a kind of a marker for a particular disease or a disorder. It can also be used for different health conditions. So biosensors, they are typically used for various applications. Now, can we use biosensor for the other application? In fact, biosensor also integrates those sensors where biological components are involved in sensing device. Now here it is very important because the recognition of biosensor is very specific and the kind of affinity they have is very high. So let us see if I have a target analyte. Target analyte could be antigen antibody, could be DNA molecule, could be substrate for an enzyme, could be a fragment of the DNA, could be mRNA, and so on. Now, what could be the recognition element? Suppose I have a substrate. Here, what you will have is you will have an enzyme as a recognition element. To clarify it, to make it more simple, I will give you an example. In glucose sensor, all of you, I think you must be aware about the glucose biosensor. In glucose biosensor, the sensing element is glucose oxidase. It's an enzyme. So enzyme is the one which is involved in recognition element. And why enzyme? Because enzyme has a very high specificity to bind. So so what will happen? The sensor, when it is immobilized or when it is coated with enzyme or it's a functionalized with enzyme, they will specifically recognize glucose molecule. It will not recognize other molecule. So that is what is the greatest advantage, and that increases the specificity as well as the sensitivity of your sensor. So you can have an enzyme where you are detecting substrate of that enzyme. So here glucose is substrate. We are taking something which is going to work on that. So you take enzyme which can convert glucose, and ultimately you have the transducer, signal processing element, and display in a particular sensor. And these biosensors could be electrochemical, optical, piezoelectrical, gravimetric, or pyroelectric. And the biosensing element or biorecognition element, I have given you example is enzyme. Another very interesting example is antibodies. So where does antibody bind? It binds with antigens. It's a very highly specific. So by looking at the antigen antibody interaction, one can detect the virus, one can detect the bacteria, one can detect the other biomarkers by using their antibodies. Second thing is that antibodies are relatively, uh, important because of the high specificity. So people they started looking at, can we have some other molecule which has a similar high specificity? So these are aptamers. Aptamers are nothing but small fragments of DNA and RNA which one can use for binding with different molecules. So these are DNA and RNA molecule. They have a specificity for binding to different material. It can bind with a protein, it can bind with a biomarker, it can bind with other material as well. Then for for detecting many bacteria, antimicrobial peptides have been used. And then the last one is MIP. It's a very, very interesting area where molecularly imprinted polymers have been created. So what is the difference between a polymer which has been created over here and the antibodies? Normally MIPs, they are called as artificial antibodies or synthetic antibodies. Antibodies, they have high specificity. You can replace this highly specific molecule with a synthetic molecule. So these are generally produced by antibodies you produce inside the organism. So they are limited. They are they can undergo the degradation. But what you can do is that you can have more stable MIPs which can be used. So for next generation biosensors, these MIPs, they are very important tool for their application. Now here I will show you some of the data which has been collected from our group or at the IIT Delhi, where we have made a sensor. This is a ID sensor, and what you have is that it's a basically impedance measurement, and you have WO3. On the surface of the WO3, we have immobilized vancomycin. Vancomycin is an antibiotic. This antibiotic have a high specificity to bind with a particular type of organism, which is Staphylococcus aureus. So why it is so? Because there is a number of hydrogen bondings which can occur between the cell wall of this bacteria and the vancomycin. In general, vancomycin is used for recognizing bacteria and killing gram-positive bacteria. So if I have this as a sensing element on the surface, if I immobilize, what it will do? It will bind very specifically to gram-positive bacteria, but it will not bind with the gram-negative bacteria. Now, some of you may ask question why this becomes important biosensor. Answer is that suppose a doctor has to prescribe that whether I should give the antibiotic which kills gram-positive bacteria or gram-negative bacteria. So you have to know that which type of bacteria, which class of the bacteria is present or is infecting. And in that case, you need to discriminate between gram-positive and gram-negative bacteria. So here I'm going coming back to the lecture. What you do is that you make the sensor, and as you may have learned about that UV visible spectra, you do it for band gap energy. You use XRD for understanding the crystal structure, whether the material is crystalline or not. Then you can also do FESEM, where you can look how the ingredients are present over there, how these molecules they are organized. Then you can do the AFM, atomic force microscopy. So you look at the atomic force microscopy, which gives you how much surface is coated with the your molecule which you are using. And other than that, you also do FTIR. Of course, one can do SEM, and then one can do FTIR. Now, FTIR will tell you, suppose you have a gold electrode and you are, putting this WO3 on the surface, we should know whether WO3 is deposited or not. So to know that you have done FTIR, you got the transmittance data, it has been got in the FTIR, which is present in our group at NRF. And then you can also look at whether the vancomycin, which is important for making a specific sensor, has been deposited over there or not. So you look at those wave numbers and you can compare that, okay, this corresponds to the vancomycin, and that's why they have been quoted. And this discrimination should occur between the gold electrode, the one which is a material which you have deposited, and then the sensing element which has been changed because of the biological material. Now, looking at that, you can see the sensor. One thing is that you can measure the concentration of the bacteria. On the other hand, you can see the specificity. Now, look at the bacteria, which is a gram-positive bacteria. So, all the gram-positive bacteria, they show at high value, whereas all the gram-negative bacteria, they are showing very low value. So, if I put a cutoff of, say, 5%, all the gram-negative bacteria will show absence, whereas all the gram-positive bacteria will show their presence. Now we are moving on to another type of device which has been made in the nano research facility. It is the one where you can develop a FET device, field effect transistors. And these field effect transistor, again, you can immobilize with vancomycin, and you can see that these vancomycin coated surface, they are highly specific for binding with the gram-positive bacteria, and that's why one can differentiate between gram-positive and gram-negative bacteria. Now, again, I'm showing you how one can characterize it using the this particular uh, IR spectrum. You get the wave number. You can characterize what is happening in the pristine sensing device, than the one where you have a coated with MOS2 and TiO2. So this way you can discriminate between the different your chip or wafers, whether they have been coated or functionalized, or they still need to be functionalization. So that means it is a powerful tool to characterize your device. Again, this is showing you the sensitivity of different sensors towards the gram positive and gram negative bacteria. Then coming to the part which I have just discussed, that MIPs, molecularly imprinted polymers, they are tool for new generation biosensors. Here I'm showing you one of the method where MIP has been made and then characterized by FTIR. So here, for example, you can take a magnetic nanoparticle, you do polymerization, and after polymerization, you can remove the template for which you have made this. And then once you do that, you have imprinted nanomaterial or nanoparticle which has been formed. Here, the idea over here is that on a template, what you are doing is that on the magnetic material, you are making a polymerization. And when you are doing polymerization, you are also having a template molecule, which is the responsible for their specificity. And once you remove the template, suppose this is the template, you have removed it. So these are the area, these are the vacant portion where these molecules can bind. So that is why it has a very high specificity. So suppose I have to make artificial antibody. What I'll do is that I will take antigen as a template and I will make the artificial antibody. Now this is the one which is showing you the difference in the spectral pattern of different IR spectra which has been taken. For example, first one, this is the one which shows that magnetic material iron oxide. You can see that at this base, the transmittance shows that just iron magnetic particle is present. Then what you have done is that you have functionalized this with MPS. You can see those bands. Further, you find that these are the one which are going to be, after functionalization, you have a surface imprinting of the molecule. So here you can see surface imprinting. So all those things can be characterized based on the type of a bond or type of the fingerprint which you have on those material.